Hi biologist, and welcome to your next screencast. Today we're going to finish chapter 9 and finish our discussion of cell division and mitosis. When we left off, we were talking about the cell cycle. Remember that the cell cycle can be broken down into the preparatory phase, or interphase, and the division phase, or M phase. Interphase can be further broken down into G1, S, and G2. The M phase can be broken down into mitosis and cytokinesis. And finally, mitosis can be broken down into metaphase, prophase, anaphase, and telophase. Let's start today's discussion by talking about the mitotic spindle. The mitotic spindle is the structure that separates sister chromatids during mitosis. It begins to form during prophase, finishes forming during metaphase, and separates sister chromatids during anaphase. The mitotic spindle is made up of hollow protein fibers called microtubules. Remember that microtubules are also key components of the cytoskeleton. Microtubules will attach to the centromere of chromosomes with the help of kinetochore proteins. The function of kinetochore proteins is to connect the mitotic spindle to sister chromatids. Most chemotherapy drugs, like Taxol, target the cell cycle and cell division. They'll interfere with DNA replication and/or chromosome separation. These chemotherapy drugs can avoid attacking normal cells by targeting the characteristics of dividing cells. Researchers have looked to plants as a source of drugs to treat illnesses like cancer. This has proven successful in the past. Aspirin, codeine, and quinine are all examples of drugs that originally came from plants. Taxus brevifolia is the scientific name of the Pacific yew tree, an evergreen tree that grows in the Pacific Northwest of the United States. Researchers found that an extract from its bark will actually kill cancer cells. The chemical extract in the Pacific yew tree bark was called Taxol. Taxol interferes with the normal organization of microtubules. It prevents microtubules from shortening during anaphase. As a result, chromosomes cannot separate and mitosis cannot finish. Cells need to tightly regulate or control cell division and make sure they're dividing only when they need to. Otherwise, cancers could occur more frequently. To do this, cells have cell cycle checkpoints. These are cellular mechanisms overseen by proteins that are found in every cell. The cell cycle checkpoints make sure that cells progress through the cell cycle properly. They ensure that each stage is completed accurately before moving on to the next one. The cell cycle checkpoints are represented by red check marks in the diagram. There are 3 cell cycle checkpoints in all. The first is in G1, and it's looking for a signal to divide. The second is in G2, and it's looking to see if chromosomes were replicated correctly and if there's any DNA damage. The third is in the M phase, and it's looking to see if sister chromatids are aligned properly for separation. As I just mentioned, one of the things that the G2 checkpoint is looking for is DNA damage. DNA damage can occur in the form of mutation. If the cell finds DNA damage, it can do one of 2 things. It can repair the damage, or if that's not feasible, it can direct itself to commit suicide. This is called apoptosis. Apoptosis is programmed cell death. It's a self-destruct mechanism designed to prevent cells from producing more abnormal, damaged daughter cells. You can see apoptosis on this slide. It's happening to the cells that look like they're imploding in on themselves. DNA damage can accumulate over time and over one's life. It can begin to interfere with multiple checkpoints. If the DNA damage affects proteins that oversee the cell cycle checkpoints, cancer can result. Cancer is a condition in which cells divide uncontrollably. As they divide, the cancer cells can form a mass. This mass is called a tumor. This table shows some of the differences between normal cells and cancer cells. A lot of the differences have to do with size, shape, and the appearance of recognizable features. The photos on the left show different examples of melanoma, a type of skin cancer. The X-ray on the right shows a patient who has lung cancer. You can see a lung cancer tumor in the patient's left lung, which is on your right. Both melanoma and lung cancer are treated with Taxol. Tumors can be characterized as either benign or malignant. Malignant tumors tend to be more dangerous because they can grow and spread more rapidly in the body. Cancer develops in a series of stages. The most dangerous stage is stage 4. This is when cancer has spread to other parts of the body and started new cancers. Cancer can be a fatal disease because cancer cells can kill by crowding out normal cells, invading other organs, and secreting poisonous chemicals. There are different ways in which we can fight or treat cancer. The first is surgery, where we physically remove the cancerous tumor. It's most effective if the cancer is localized or only found in one place. It's not effective if the cancer has metastasized. To metastasize is to spread to other parts of the body. The second technique is radiation therapy. In radiation therapy, high-energy radiation beams are focused onto a tumor. This kills dividing cells by damaging DNA. Because the DNA is damaged, apoptosis is triggered in these cancerous cells. The third technique used to fight or treat cancer is chemotherapy. Chemotherapy is the use of drugs that interfere with the cell cycle and cell division. Remember that Taxol is an example of a chemotherapy drug. The photo on this slide shows a patient undergoing chemotherapy. These graphs show the effectiveness of Taxol. The graph on the left shows that ovarian cancer patients live longer on average when treated with Taxol. The graph on the right shows that breast carcinoma patients have less recurrence of the disease when treated with Taxol. Taxol is highly effective, and this has caused challenges. The demand for Taxol has increased dramatically. This has increased pressure to remove Pacific yew trees from forests. The spotted owl lives in these forests, and deforestation threatens their habitat and the survival of their species. One solution is developing synthetic Taxol so that we can produce Taxol without cutting down these trees.